of secondary metabolite. The first one is intracytoplasmic storage type, and the other one is extracytoplasmic storage types. So the intracytoplasmic types are molecules are accumulated in the vacuole or cytosol or the other organ. It means inside of cells, the secondary metabolites are accumulated in this type, and extracytoplasmic storage types are secondary metabolites are binding to the cell wall or secreted to the medium, not the actual medium, the outer of the cell. You can say that. So we have to think maybe the type of our target metabolites are intracytoplasmic storage type, then we can use cell culture type. The target metabolite is extracytoplasmic type, then we have to use cell culture type, because if we our plasmic type, but if we choose cell culture, then all the secondary metabolites are is go outside of then the metabolites are available. So this is the apprenticeship routes are more safe way to get the target metabolite. So the last time it is around compare the cell and organisms culture system to produce target metabolites. That time our material was Panax folios, which is from originated from USA, America, and the other one is Panax gene. So we used four cell lines from two species. I will compare the cell and organism root types. So as you can see here, from all cell types, their cell produce more fresh rate. But if you see the dry rate, the adventitious roots are higher than cell. So you can see the biomass always adventitious produce more biomass than cell. So cells are full of water. If you look at the secondary metabolite, this is the ginsenoside content. And cell contains a lower amount of ginsenoside compared to adventitious roots. And adventitious roots, not only the content, their composition of the ginsenoside are also more than cells. Cells are more simple in terms of secondary metabolites. And this is the also we compare the cell and adventitious culture system again from using other four species of plants. First one is Camellia japonica, Acetala sciatica, Euripoma, Morinda. Those four species we use to compare with those two systems. And here, interestingly, the result was a little bit different with the previous one. So here, the cell show the contents of total phenolic compound. They show the cell. In here in Camellia, they show more total phenolic compound than the phenolphthalein. But in case of other systems, they show the root produce more. So it depends on the species sometimes. But in case of ginseng, the adventitious root system was always better. But the other species is shown slightly different. It depends on the species. That makes our study difficult. There was no standardized system. So here also the composition of phenolic content. The cell show a high amount of in here Camellia, and in case of Morinda, they show the high composition in cell, high composition of the phenolic compound, but less in the adventitious root. But the other species were different. And this one is the I'd like to introduce about tissue-specific compound production. For example, I saw, it was very interesting for us. Central Asiatica could grow outside. So we found that in front of the Duitan University, just in front of the university, and the ground there was a Central Asiatica. But in Korea, we have to keep them in greenhouse only. We cannot grow outside. That much it was, but the Korean company, like a cosmetic and pharmaceutical company, they are really interested for Central Asiatica. So we also, but the interesting thing of this trend is that it can be used in many products, especially one of the huge pharmaceutical company, Dongbu Je약. They are one of the major compound is the Central Asiatica, and almost 100% of Central Asiatica used in that company. They imported from Madagascar, which is a huge, famous island. So they want to get some more Central Asiatica from other places instead of Madagascar. So we also have a collaboration with them. That's why we studied first about this. Their major compound was trichlorophenoid, saponin, maracaside, asiatic acid. They never consider other metabolites. They only consider these four compounds. So, but the interesting one of this plan is around 80%. Around 80% of those compounds was founded in legally, and there was petiole, root, node. The other part, less than 5% of metabolites are accumulating. Coral leaves, they contain high amount of asiaticoside. It is the most popular among that. But as I told you before, our system is adventitious root system. But from here, the root, 0.17% was produced from root. So we were so much worried. How much? We already have a collaboration with the company. We have to produce lots of, we have to show some results. We can produce lots of metabolite from our system. So how can we produce? So we didn't know that time first. So we use many systems. But first one was the agrobacteria. We use agrobacterial rhizogenes to induce hairy root. So the explain was cultured in greenhouse, and then in vitro plan we introduced, and after home culture with the agrobacterial rhizogenes, we used to give it after 2 weeks. We can hairy root from there, and we culture like the nicely and compound using PCR. The genes are all genes are inserted in there, and then we keep propagating them. So our system was hairy root system, but that time we induced the hairy root from using two types. The first hairy root were induced from leaf, because leaf contains a high amount of trichlorophenoid. But petiole root, they didn't compound a lot. So we induced hairy root from those samples. But as you can see here, this is in vitro plant. In vitro plant produced around, in vitro plant, you can see here, they contain 4 types of trichlorophenoid. But compared to that, in vitro advantage, the hairy root, they have a very simple, they contain more than in vitro plant, they contain secondary trichlorophenoid. But the composition was very simple, like only one type. And the other thing is that actually leaves, the original leaves, explain they contain high amount of trichlorophenoid, but the hairy root from induced from leaf or petiole, the hairy root from the petiole, they produce more than leaf of originated value. That means the original explant doesn't give an impact to the value. It's just depending on the selection of the line is important. So that's what we obtain from there. And after we get some kind of material, whether it is a helium or adventitious root or canvas, then we have to increase the secondary metabolite from there. So usually the secondary metabolites are increased when plants are affected by insect or fungus or kind of bacteria, and the other one is when people are manipulate the biosynthetic pathway of the target metabolite, then we can increase the secondary metabolite. So the rest of that is the how to we can increase. So I want to tell you about that. So there is a simple way to increase the secondary metabolite. The first one is precursor feeding. So if our end product is this food, like as I told you before, this is central asiatica, the four main metabolites are asiaticoside, madagascar, asiaticoside, this kind of what tryptophan is important. Then these are pathway of the metabolic compound, target metabolite. Then precursor feeding means we can add in the medium those derivatives. But there is a one rule. If we want to feed some precursor inside, then precursor should be cheap. Because other one is very expensive. So we that time we were thinking which one is cheap. Very cheap. It should be very cheap. So the squalene was the cheapest one. So we can this much of a tablet, around 500 of tablet, we can buy it in cheap price. So we use squalene for feeding. Uses precursor feeding, and the other one is elicitation. So people there's a kind of using plant defense mechanism they are using it. So chemical elicitor and many kinds of elicitor we can use to induce defense mechanism. So here is our result around. Sorry, this is a little bit old, but anyway, when we use elicitation in ginseng adventitious root culture, we use methylxasconate. So here, the squalene at 40 days expression was high, so we can get some increasement of ginseng side. And this one is the precursor feeding. So by using precursor feeding method, we put squalene inside. So after that, we can get some. Previous time I told you about in case of Central Asiatica, and this one is ginseng, but their whole target compounds are three terpenoids. So this one also precursor. As a precursor, we use the same one squalene we could use because they are the same three terpenoid. So here, when we use squalene for precursor feeding, we can get some higher amount of final product. And also, when we check the gene expression, like squalene gene days and squalene epoch days, also we can get some higher expression of genes. And this is the other way to increase the secondary metabolites. Some of the students, some of the audience, they already saw the day before yesterday when I had the presentation in MNMP 2026. That time I presented the mountain ginseng adventitious root culture system. So one of our oldest mountain ginseng adventitious root culture was maintained 25 years. So it was old, and sometimes the line produced very sometimes low, decrease the amount of ginseng. So we need to renew of the ginseng. So this experiment was one of my excellent students from Vietnam. Her name is Kim Hoon. She is now working in USA as a doctor. So this experiment was done by her. So that time to get the mutant line which produce a higher amount of ginseng side. We tested the radiation breeding technology to induce the mutant. So we treat the gamma radiation to the plant, induce the mutant. So we obtained tons of adventitious roots. Very difficult to select because we cannot test it using HPLC. Thousands of materials are there. How can we? So first we selection is that whether the adventitious root produce a high amount of ginseng or not. First, they have to produce lots of biomass. That's the first. So first selection, the guideline was they must produce. They have to propagate a lot. That's the first selection. So we selected more than 15 lines according to their biomass production, and then we culture them again, and then second selection was by ginseng content. So we select again because those all cell lines are proliferated well. So in second selection we total ginseng content we analysis, then we selected again. So we got the really line from the older line. We checked for the publication, that one was not enough. So which kind of gene was mutated by gamma radiation? So we did some molecular analysis too. So anyway, so we found some genes was manipulated by gamma radiation, so that we selected the line to produce more ginseng than previous one. And the other one is the hair removal culture and genetic system. We could produce more amount of the target metabolite, which is ginseng. But actually, hair root and transformed roots are actually GMO, considered as GMO. So it is difficult to directly go to the industry. It is not possible in Korea. But anyway, this is also good way to increase the metabolite. So this is the agrobacterial rhizogenes. If we culture agrobacterial rhizogenes to the plant tissue, that is one of the role gene was integrated to the plant genome, and they can produce this kind of keep proliferated root. It is called as the hair root. And the other one is the agrobacterium innovations. And in here, in vector system, we have to put some our interesting genes. Our target gene should be inserted, and this vector delivered by agrobacterium innovations, and it goes to the plant genome. So in two ways also we use, for produce more ginseng. So this is the pathway of the ginseng. So here we were interested in superRNA synthase gene and superRNA oxidase gene. So we introduced to the our mountain ginseng adventitious roots. So we got how many? 6 lines we got. Anyway, I didn't bring the data here, but anyway, we could produce more amount. But it couldn't go to the market directly because it is NMO. And I want to introduce about the large scale culture for the use of plant nutrition culture products. So I knew the Dr. Tam also doing nicely in here for the bioreactor culture. There is a conventional culture method. This is a petri dish, as you all know, it is around 9 centimeter diameter size, and this one is bioreactor. So air-reactor type bioreactor. So the petri dish size, small scale size, we can say that small scale size is around 10 to 500 ml of medium is there. A bioreactor usually start to read the 1 liter scale to 10 ton scale. 10 ton means, you know, the around 10 ton, 10,000 liter. Okay, so the petri dish is easy to handling, and no need specialized facility, and partial contamination is easy. We can just get rid of them. That's all. But the bioreactor, we need a special techniques and need a special facility like Python bench, big bioreactor, one time contaminated, then we have to do all of them. So that is the not good one in bioreactor. But if you see the petri dish, if you think about the industry, then the petri dish is okay for the research purpose. But if you think about the market industry, then petri dish we cannot produce that much of biomass. So rigorous work with many hands, we need a lot of work for the cell culture. But compared with the petri dish bioreactors controlled by automated system, it is a half automation and easy to scale up the factory. It doesn't need any agar or Vitazer, that kind of solely, it doesn't not necessary. So the cost is down, the medium preparation and the reduced production cost. As I told you before, we don't need any solid gel. And controlled quality batch is easy. So almost all materials are controlled. So the ingredient is also safe. Easy to scale. That's the most good one. And there is many various types of bioreactor is there. So these type bioreactor we often use, frequently used. So as you can see here, the areas are goes from the bottom to this ladder, which is flat. There is a small holes on the side. So all the cultures, cell and adventitious roots are lifted by air. So this time the oxygens are very important. But that oxygen is one of the main factors to increase biomass. And if we supply the air through this funnel, then outside of air should go outside. That's why the unwanted, the hazardous gas can be removed from the bioreactor. That's one of the reasons to cell or adventitious roots are increased more than calculations. So here is the filters, it can be sterilized through the filter and go inside. So, and deal, we can put the deal and everything. It goes like this, and we can control the air, how much of air, we can control everything, and then we can just dissolve the oxygen and the pH, we can monitor. This one is the petri dish one. So in case of petri dish, compared to petri dish, the bioreactor, you see here the growth index is 2.7, but this one is 20%. So the increased by 10 times increased biomass we could obtain from the bioreactor. On this one, we got this result using Angelica adventitious root cultures. And first, when we culture adventitious root, first air root adventitious root numbers are increased first, and then once adventitious root was induced, then the length increased, and then adventitious root number was also secondary. The adventitious root also increased. So we can see this kind of graph for the biomass. This is the dissolved oxygen. How increased how their curve are changed during the culture system from 0 to 4 weeks. First time dissolved oxygen was high, but after 1 week, 2 week, third week, it goes down because of they grow a lot inside, so they use a lot of oxygen maybe. So after seeing this kind of graph, we thought that okay, this time maybe they are run out of oxygen, that we need to supply more oxygen there. So usually third weeks we put more oxygen so that they can use. Actually, this one is the magnified previous graph. What I obtained from the dissolved oxygen monitoring. First, they do not. This is kind of because of the respiration. The adventitious root also respirate during the night and day because they are vital adventitious root. So first time there was no big differences. Second day, third day, fourth day, there is no big differences day and night. But if we see 16 days, 17 days, 18 days, then their respiration is very different. So that I thought maybe there is a circadian kind of rhythm is there, and they respirate maybe affect to the biomass also. But I couldn't do deeply about. I couldn't study deeply about here, but I wanted to do. But anyway, it was interesting. So this one is the bioreactor cell culture one, and that one is the plant culture in the bioreactor. So depending on the target metabolite, we could choose what kind of system we are going to use. So this is a small scale, like bioreactor. So the previous bioreactor was air-liquid bioreactor introduced to get some biomass like a cell and adventitious root. And I'd like to introduce about the temporary emergent bioreactor for plant production, not a biomass like a cell or adventitious root. So the plants we need to send simple. This is the three kinds of temporary emergent bioreactor systems are there. The first one is two-butter system, so that we have a two-butter. So there is a solenoid valve. So solenoid valve attached with the timer. So according to the timer, it can be closed and open, so that medium can go inside to the plant, and after that, after some time, it can go outside. So it moves. So that is the two-butter system, and the other one is gravity draining system. So using gravity. So first by pumping, this one goes inside of the bioreactor. Then after some time, solenoid valves are open. Then by gravity, it can go automatically down to the water again. So this is the gravity draining system, and the other one is the internal medium reservoir system. So reservoir is here. So after pushing by air, the medium can go upside, and after some time it goes down. So it is kind of temporary immersion for plant growth. So many types of bioreactor, temporary immersion types of bioreactors now you can buy through the internet. So this is the CETIS, and the Europe people used to use that one, and the company may be located in Belgium or something somewhere. Now I forgot. And the other one is Nalgene and Matis, Rita. Rita is small, and Matis also a little bit bigger than Rita, but and you can see there is a two-butter system used some other laboratory. This one also two butcher system. And the plant home also we can purchase from abroad. But in our case, we are using septic systems. But that one is made with plastic, so we can after play, we can use several times. Only the water, so this system is there is a port for the medium supply and air supply. So that portion is a little bit big, so easily breakable. And one more thing is that they are a little bit expensive because it is a European product. And this one is actually before using that one, before using the European TIB system, we use the air with bioreactor for producing the plant. This one was Professor, I forgot his name, his full name, because he was our friend, close friend, the Cat, Professor Cat. And he already retired from Nala University as a high school chancellor. So he retired. So when he was a PhD student, he did this experiment. It was quite long data obtained from him. So this is the temporary immersion bioreactor system using Anectocellus chromosomes. So there is four types of system. We can put the medium inside, and then after sometimes it can drain or rotate. And from those systems we could obtain, you can see how much of plants we could obtain from there. So this shows the plants inside. And this one is the kind of small culture vessel. From here, we can get around 25 to 40 plants left per vessel. But from the bioreactor, there is a bioreactor. Every small things are shoot up. So from here, we can around 1,000 to 160 plants left per 5 liter bioreactor. So you can see how the system is useful. There is after 2 months of acclimatization, we can get some useful acclimatized. You see very popular person here today. So this one, it looks younger than, a little bit younger than now, but after so he wore the 100 years old polygonal multicolor tube. So that time we had a collaboration with a big cosmetic company. So they are very particularly, they want to please produce the product from 100 years old. They said it is very good for marketing. So for us as a scientist researcher, for us whether it is 1 year old or 100 years old, it is actually for us nothing. Whether it is produced a secondary metabolite or not, that's the important for the researcher. But the marketer, who is the company people, for them, this kind of is very important. What kind of material to use, because can be a good story for the marketing. So anyway, we got a very expensive material we obtained, and then at that time Dr. Hotanta was a really good researcher. He was the candidate student, so he got the material, and then he produced several lines of polygonal multiforum. And according to scale, you can see here, we started with the 0.25 liter, so we tested 500 liter, 0.5 ohm. So according to that, if you see the growth index, the 3 liter bioreactor, they produce maximum biomass. If we go the high, if we increase the volume, like a 500 liter, then the growth index slowly decrease because of gravity and then air curing stress, because of so this one and Dr. Hotantami published many publications before before he graduated. And so this is the pilot scale bioreactor. It is the 1,000 liter bioreactor. So before harvest, we have to put some elister. So here you can see some elixtation. We use elister methyl but and here you can see the elixation port is here. So through this port, we can add in elister there, so that we can finally get the good quality of the biomass from the tank. But for the we commercialize advantageous route through this large scale. But there is actually lots of problems are there. I think here the young students are here, so you guys can solve those problems. So one of the bottleneck, I can say the bottleneck instead of the problem, that is methyl jasmonates are really expensive. So if some the future researcher can find the substitute or alternatives instead of methyl jasmonate, put some magic chemical there and increase more secondary metabolite instead of methyl jasmonate, then it will be very useful. But right now we don't have no options. We have to use the expensive methyl jasmonate inside. And the other bottleneck is that the cells there is a status of cell. There is many different types of physiological states are there. This is the protoplast. There's small size of protoplast. Cell types are during the cell culture process. Cell types are there is undifferentiation, redifferentiation, differentiation, redifferentiation. Many types of physiological status are there, but we try to change and we try to derive our direction of the cell type, but we cannot drive as we want. We try to. So that's really what we want to do. So that's also, I'm trying my best, but I cannot solve all the problem. So I think you guys can do that. The rest of my homework. So my slide is I brought this from. This is maybe taken around 8 or 10 years back. Oh, 8, 7 years? 6 years. 6 years? Oh, really? You can see the donut there. Oh, yeah. 2029. I'm easily forgot. 7 years ago. So when we all the land members, when we went to the membership training during the summer season, so almost all the students are graduated. Many of the students are married and they have a family already. That time she was a baby, but now she was 9 years old. So it was a very happy time, and I still remember how we were happy. So those kinds of happy day makes our future now. And thank you so much.